Recording in progress. So, who is Davide? Well, he is a clinical toxicologist at the Centro antiveleni and the Centro Nazionale di Informazione Tossicologica. You can hear that I don't speak Italian, and they are part of the Institute of Clinical Science in Malgeri in Pavia, and that's a city south of Milan. Davide did his medical studies and his habilitation at the Università degli Studi di Pavia, and he also specialized in medical toxicology at that same institution. Now, as a medical toxicologist, Davide works with patients who contain things that they should not contain. So, for instance, high concentrations of cobalt or mercury, snake venom, intoxicating opioids, botulinum toxin, or even alkaline batteries. He has an interesting series of papers on a number of inclusions that you should not find in patients. Now, today as well, we are getting very close to the summer holiday season. I don't know if it's the same in your countries, but here in the UK, everybody is traveling. It's half term here, so the airports are busy, but everything has been effed up. So I had a friend visiting this weekend, and he was supposed to have left on Sunday, but he only left last night, and then he got stuck in Belgium, and only today could he manage to get back to the Netherlands with 3 days delay. So everything is upside down. But people will, in this holiday season, of course, get into close contact sometimes with dangerous venomous creatures. And considering the audience, probably some of you will do this on purpose. So Davide's talk today is about what will happen if that goes awry. The title of his talk is Venomous, Bite and Stings, the clinical experience of the National Poison Center in Italy. So Davide, thank you so very much for joining us today. I'm happy to host you, and I look forward to your talk.
Thank you very much. Good afternoon to all. It's a pleasure for me to talk about our experience about envenomations from a clinical point of view, but we need all expertise around the world to know better the venom. And I should start with a short description of our unit. I work in Pavia in a poison control center. It's one of the poison centers in Italy, and with some particularities that the activity of poison center in Italy is a nationwide activity. We have no geographical or regional competence. We have around 80,000 calls per year. It's very much. It's open. Our service is 24 hours a day, 7 to 7. And an important characteristic is that the advice is only by clinicians and expert in clinical toxicology. We have a front office urgent line service and a back office activity that's very important. And we need to work with the biologist, technician, chemist, pharmacist, and administrative personnel. Our service is the unique in Italy that is the point of reference for government and institutional organization. We work for the Italian Department of Anti-Drug Policy, for example, for national activity of new psychoactive substances. We are the reference for the CBRN reference center from Ministry of Health and for chemical accident. Now, today we talk about the 2% of our activity, because as you can see here, the 45% of our activity include drugs. The other 27% household product, and 2%, but is important to know and to better manage the patient envenomed or bitten by venomous snake or spider, for example, or other animals. And we can start with the most frequent envenomation in Italy due to snake. The unique venomous snake in Italy is the vipera, and although the Europe cannot be considered the dream destination for herpetologists or clinical toxicologists, but we have in Italy, for example, 4 different kind of species of vipera. We have the Vipera aspis, the Vipera modites, Vipera berus, and the Vipera ursini. The venom of this snake differs, is a mixture of proteases, metalloproteinases, and other compounds that cause local and systemic effects. Disorders, hemodynamic disorders, coagulation disorders, and local tissue necrosis or complication. And a particular aspect of the venom that we studied here in Pavia is the neurotoxic effect of venom snake envenomation. Then, from a clinical point of view, we use a score to grade the severity of envenomation. We have a score including 4 different stages. The grade 0 is the initial sign of bites. More than 50% of cases remain grade 0 and are defined as dry bite. As you know, the snake may bite, but the snake don't inject the venom. And the grade 1 is characterized by only local signs, local edema, without systemic symptoms. And the grade 2 is characterized by a regional edema, a progression of local signs associated with general symptoms like hypotension, vomiting, or diarrhea. Then the late stage is the grade 3, is characterized by regional edema or severe systemic sign of envenomation. And as you can see here, the clinical manifestation. We know that we have an antivenom, or more than one antivenom, that we can use in this patient. And we know the medical literature is agreed with the. There is a sound in the background. Okay. The medical literature agreed with the antivenom administration in cases with grade more than 2, and is characterized then from an increase of local signs associated with systemic envenomation. Here we described a little baby that was bitten by a Vipera aspis. 30 minutes after the bite, presented with the local edema, and after 20 hours, the local edema was associated with ptosis and neurotoxic effect involving only the cranial nerves and characterized mainly by ptosis and deficit of masseter muscles, for example. The antivenom was administered, and 72 hours after the clinical manifestation resolved. Okay, then we studied from 2001, 2011, our experience in our center about the neurotoxicity. That was an aspect few known and few described, in particular about the specific mechanism of toxicity of some venom of some species of vipera in Europe. We included 24 patients. These are the case series more numerous in medical literature. We included 24 patients with 80% adult and 5 patient pediatric children, pediatric patient, and the clinical manifestation are summarized here in the slide. Included the visual complaints and ptosis and ophthalmoplegia as a more frequent neurotoxic effect described. As you can see here, we divided the the case series described in pediatric group and in adult group, and about the treatment, the antivenom treated. As you can see here, the patient in pediatric group was all treated by antivenom, and in adult group, 19 of 23 were treated with the antivenom. But an important aspect is summarized in this table, because as you can see, the neurotoxic effect in a red cycle was in more than 5 patients, because we have 1, 2, 3, 4, 5, 6, 7, 8, 9 patients, was the unique expression of systemic toxicity. And in pediatric group, all patients presented with extensive local signs. Okay, in adult group, most of patients presented with neurotoxic effect as unique systemic manifestation of envenomation. This clinical manifestation was correlated with only local signs. This is an important point because these aspects may modify the clinical approach at the need of antivenom. Then we proposed and modified the grading severity score suggested by Audebert and other French authors in the last years, and we modify this clinical grading, including the manifestation of neurotoxicity, and consider only the patient presenting other than ptosis and other neurotoxic signs, including ophthalmoplegia, dysphagia, or other more severe signs, patient that may receive an antivenom. The patient presented only local signs associated with only ptosis may require an approach, a conservative approach, and an approach of medical staff is suggested to wait and see and not administer antivenom. Then we studied, and this is your field of research, because as clinician we don't work. We work sometimes with researchers and experimental study, and we studied in our snakes in Italy the venom. This is the first tentative to milk these Vipera aspis. As you can see, the bottle is not adequate, and the quantity, the amount of venom that we received from a snake, from Italian viper, is only 1 drop for a fang, and a very little quantity that require appropriate tools to store this venom. And as you can see here, we ameliorate our tools to store the venom and to analyze this with the specific and essential help of a team of researchers of University of Padua, coordinated by Professor Montecucco and Professoressa Ornella Rossetto and Marco Pirazzini. We studied the composition on the Italian Vipera venom, and in this biochemical characterization via plasmapheresis, we try to understand the composition of the venom in this tool. And the important message in conclusion of this experiment is that in the Vipera berus venom there is few compound with 14 kilodalton molecular weight. And in contrast, we have in Vipera aspis a very important amount of proteins that probably could be phospholipases A2 that play an important role in neurotoxicity. After this first experiment, our colleagues studied the activity of the phospholipases A2 in vitro enzymatic assay, and an important result is that Vipera aspis showed an important and very high phospholipases A2 neurotoxic effect compared with the B phospholipases A2 used as standard in this experiment, and Vipera berus don't show any phospholipases A2 activity. This is an important point because the clinical manifestation after Vipera aspis and Vipera berus bite may change and may require different treatment. In vitro neurotoxicity showed an essential and important changes in these motor neuron cells incubated with the venom of Vipera aspis in this case. And if you see here, you noted that the modification of the plasma membrane and the formation of bulges. These bulges are due to this mechanism that we identify. This is the cycling and recycling, or at presynaptic level, of acetylcholine vesicles. Normally, during a physiological pathway, the vesicle recycle, release acetylcholine induced by calcium and other neuromediators, and then restart and the release of acetylcholine. Phospholipases A2 contained in some snakes, but also in Italian viper snakes, modify the structure of the vesicle and cause a blockade of the recycling. Then the membrane remain in this position with a fusion with the cell membrane, creating an increase in space and determining the formation of bulges, as you can see in the previous essay. Then we follow other experiment in ex vivo electrophysiological recording in vivo poisoned muscles of mouse, in particular with the injection of the venom in the soleus muscles. And then at 48 and 72 hours, we registered the potential of depolarization cell membrane in different, due to different venom from one end. On the other end, we studied the structure of the in vitro studies of the motor neuron cytoskeleton. We studied the effect of the venom on acetylcholine receptor as indicator of postsynaptic damage. We evaluate the activity of the one of the protein included in the snail protein complex that is characterizing that characterize the presynaptic activity at neuromuscular junction, and then another tool that merge two aspects. And as you can see here, there is a big difference between Vipera aspis effect and Vipera berus effect in neurotransmission. Then the Vipera aspis cause neuroparalysis and Vipera berus no. At the same time, Vipera aspis cause a damage at presynaptic level, but not at postsynaptic level, because the acetylcholine receptors are normal, and here the activity of a marker of presynaptic activity of the snare protein is completely destroyed. And these are 48 hours, and the 7 days, the process is reversible. This is another important information that permit to conclude that the phospholipases A2 activity contained in the venom of Vipera aspis in particular is similar for the clinical point of view to the botulinum toxin. Okay, because the mechanism of botulinum toxin is different, is the poison of the poison, because it's very potent. It's the most potent natural venom on the world. Okay, but the mechanism differs because the botulinum toxin cuts as a scissor the component of the snare protein that include and permit the fusion of the presynaptic level of the vesicle containing acetylcholine, but from a clinical point of view, the clinical effect at the end of this process is the same. To date, the effect of venoms in Italy, including neurotoxicity, is limited only to cranial nerves, probably due to potency of the toxins, and probably due to also the activity of the cranial nerves, for example. This very high activity requiring a very fast and continuous restore of acetylcholine, because require a very important turnover of acetylcholine. We tried also to evaluate the antivenom activity that may counteract the toxic effect of venom, and as you can see here, this antivenom, one of the antivenom available, is not able to counteract all the activity of the toxic venom, and is not the best choice in this moment. From an experimental point of view, as you can see here, the Vipera aspis venom directly injected in the muscular apparatus create the Vipera aspis no major evidence of toxicity. And for example, Vipera berus, the effects are characterized by an important hemorrhagic effect that is not apparent in clinical, in human envenomation. And this, in particular, this effect is counteracted by antiserum, by antivenom. This is the same animal that received venom, and in this, the same animal received the venom of Vipera berus, but in one case, normal immunoglobulins as control, and on the other hand, the antivenom that counteract completely this particular effect in animal, not present in human envenomation, but is an important message. All these results are described in detail in this important paper that we published in 2018 in an important paper. Okay, then we thought to better understand the activity of some venom of Italian Vipera snakes, but we received a call from Bulgaria, from colleagues in Bucharest, that asked for an expert advice about the neurotoxicity appeared in a young baby bitten by Vipera berus in Romania, associated with local mild signs of envenomation, but with neurotoxicity. And in a country, in Romania, only Vipera berus is present. Now, probably we need we need as clinicians your help and researchers help to better understand the composition of the venom in different countries, in different areas of Europe, because the venom composition inside the same species may differ, probably due to interspecies changing, hybridization of species, and different other reasons. Probably we need to better understand also by proteomic study probably the genetic reasons of this phenomenon. But about the antivenom that we have in Europe and in Italy, we have more than one antivenom that we can use. This may be a problem because some antivenom are available from sometimes and then disappear. Some antivenom are available only in some country and not in other, depending on availability of the manufacturer to have this antivenom also for other countries and not for ourselves. And we have some antivenom, for example, Viperatap, that are very expensive compared to the other one. We have different problems about antivenom. One is the different host animal used and the different composition, because most of these antivenom are from equine. Only one is from sheep, for example. Most of them are fragments, but as you can see here, the specific activity against the subspecies of Vipera differs. In Italy, we have, for example, in all over Italy, except for Sardinia, we have Vipera aspis, and Vipera berus is limited to Northeast Italy. But the antivenom available are have a specific activity against the Vipera berus. This is a problem because most of envenomation in Italy are due to Vipera aspis. Okay, there is a problem about the route of administration registered by manufacturer, because we use this antivenom as off-label, out of indication, because all antivenom should be and must be administered intravenously and not intramuscular or subcutaneous as registered. We have a problem to understand the capacity of neutralizing activity of every antivenom, because, for example, the unit that we use to compare is different. And the last is the suggested dose is very different from different antivenom, from 1 vial to 2 vial to 5 vial, depending on different product. Then we studied the performance of 4 different antivenom during 6-year study, and we enrolled 114 patients, and we can go to the results. As you can see, the most of patient was admitted to emergency department as GSS 1 or 2, then with only local signs of envenomation, or GSS 2, mild local sign of envenomation associated with systemic toxicity. And then most of patients receive antivenom, and according to medical literature, as GSS 2 or more. Then some patients presented also neurotoxicity associated with gastrointestinal symptomatology or hypotension. But this is an important message. As you can see, during this 6-year study, we use 4 different antivenoms. Due to the different availability of this antivenom in different emergency departments all over Italy, and the quantity of antivenom administered depends also on amount of antivenom available in every emergency department. We used mostly Biomet is one of the antivenom, and in 31 patients we use product manufacturer in Zagabria, Piperactab and EUVEN. As you can see in this figure, the blue bar showed an improvement of the patient after the antivenom administration, compared with the orange bar that showed a worsening of the patient after the antivenom treatment, evaluated after 6 hours post-administration. We note that EUVEN, Piperactab, and Zagreb showed a very important improvement after the antivenom administration, compared with Biomet. 50% of patients after 1 or 2 vials of this antivenom worsened also after the antivenom treatment. This is an important point because this denotes a little activity of this antivenom to counteract the venom activity of our Italian vipera. In this slide, you see the different formulation and different name of different manufacturers of antivenom available in Italy and in Europe, I think. And this is one that this is a new product that is under study and not available commercially at present, but probably in the next future. This is a polyvalent product, and we evaluate all the characteristics of different antivenom in this review published in Clean Tox that describe some of different aspects that we previously evaluated, but in this table we tried to evaluate the median length of hospital stay in treated patient, in severe poisoned patient, in severe envenomed patient, and the result is this: is that patients that not require antivenoms in this group, the length of stay was more longer than patient required antivenom, and this is the first result. The second result is that the patient received the antivenom intravenously. The length of stay was shortened than in patient that received some antivenom intramuscularly. Then the correct way of administration of this antivenom remain intravenous, and there is evidence that the treatment with antivenom reduce the complication in envenomed patient and reduce the length of the hospital stay. Because without antidote, without antivenom, the complication appear, may complicate the clinical course of the patient, in particular of the pediatric patient. We need antivenom. We need to evaluate closely and monitor the follow-up the patient very close, because the compartmental syndrome that is may be frequent in other envenomation by other snakes in Italy is and in Europe due to viper envenomation is not frequent, fortunately. And if the patient is treated accordingly to medical literature and in time, we avoid and we reduce the probability to have a complication as well as described in this case. We work to ameliorate the availability of antivenom and the antidote in Italy according to Ministry of Health, and we have a database, for example, free in our website that include the stockpile of every emergency department on intensive care units or pharmacy in hospital, and in case of need we can find the antidote in the nearest hospital, for example, and to ask for urgent supply of the antidote if needed. Then I finished my first part about snakes and viper envenomation. Please tell me if I have some minutes, or we can
so
open.
Davide, how many minutes would you like? You have had,
as you know,
37.
As you know, I can talk for hours. No.
What would be a natural break for you? If you want to do another 10 minutes or so, that's fine with me. But then we have some time so we can finish within the hour with some questions.
Sure. Okay. Just because another important aspect is the spider bites. In Italy, we have only two venomous spiders. We have Loxosceles rufaciens, not reclusa, and Latrodectus tredicinguttatus. But from a clinical point of view, the difficulty is to better understand, to know, and to correlate the cutaneous clinical manifestation with a possible spider bites. As you can see here, the patient arrived at the emergency department with the local edema, hyperemia, and other effects, but in some cases, it's possible to identify correctly the spider. We use support of experts, as well as herpetologists. In this case, we ask for expert identification by arachnologists through the telemedicine. That is, the Poison Center is the first example in the history of medicine that we consider as telemedicine. Okay, and we send the picture of the spider to the expert, and this for example, in 7 months, we try to evaluate this performance. In 7 months, we managed 50 spider bites, and in more than half of cases, the identification of the spider permits to discharge a patient from the hospital, or maintain the patient at home without the access to emergency department. This is an important role of Poison Center to save resources and to correctly manage these cases. In Italy, since 2 years, we have a tsunami of call about Loxosceles and rufaceous, ragno violino in Italy, fear, that require also some warnings in our website. The ragno violino, the Loxosceles rufaceus, is related to the Loxosceles reclusa. It's a very venomous spider that may require surgical approach and specific and pharmacological treatment, for example, to avoid local complication, and is characterized by local effect and rarely cause systemic effect. This is one point. The other spider is the Latrodectus mantas. Is a malmignata in Italiano. Is characterized by 13 red point on the body of the spider, and for the first time, 2 years ago, we used an antivenom in for the treatment of the patient, adult patient that presented with only mild local signs of sting, of bite, but with a systemic effect characterized by abdominalgia and neurotoxic effect. In our poison center, we have an antivenom that we send to the south of Italy because this patient was located in Puglia, in the south of Italy, and with an urgent supply of the antidote, we treated this patient. I have a video, very short video to show you and to share with you that
no, è che c'ho i bambini. Quindi sono scatti praticamente.
Okay.
Di breve pesce.
This is our Italian colleague that talk about the clinical manifestation of neurotoxicity, pathognomonic for this envenomation that include abdominalgia, acute abdominal surgical abdomen, and neurotoxicity that manifested with tremors, spasm, and we use the antivenom, this antivenom, Latrodectus matans, product by Merck, okay, and is one of the antivenom used in these cases. We have used this with a prompt resolution, immediate resolution within 1 hour after the administration. Is very exceptional response to the antivenom. As you can see here, we compare 3 different products. This is the cheaper product with a high performance of the activity. This is another product that is more expensive, Aracrim Plus, and this is another product, the redback spider antivenom product, manufactured in Australia. None of these antivenom are specific for Latrodectus tredicingutatus, but work well against the venom of Latrodectus matans, vedova nera in Italia. But our experience is very positive. Then I can finish. I can stop here if you.
I think that would be a good point to stop, Davide, so we can take some questions. Some have come in the chat.
Okay.
So do you want to have? Do you have an acknowledgement slide, or will you stop here?
No, we can stop.
All right.
We can stop.
In which case, rock and roll. Thanks very much. That was interesting and also worrying. I mean, to see. I'm not a clinician. All the challenges that you have. It's not straightforward when you can choose from different antivenoms and they don't work the same, and you need different quantities. It is that sounds to me quite worrying. It must be quite stressful for you as a clinician to balance these decisions quite quickly, because these are urgent cases, I guess, when the people come in. Okay.
Yes, we study for this.
Yes. Yeah, well, thank God. Let me start at the top. Mike Dam asked a question. He says, when you use the Vipera berus and the Vipera aspis venom in your experiments,
okay,
are you using a pool from different populations, or do you use venom from one population?
Yes. Is a good question, because we are studying, because we are searching for grants and for a project, because my thanks to our colleagues in Padova is also about this aspect, because all the experiment was done without any grants. And many thanks. But in this field, we collect and we milk some Vipera only in 2 different regions. One in Lombardy, near Mantova, and in the northwest of near the Piedmont, and for Vipera aspis, and for Vipera berus only 3 Vipera berus of the northwest near Bolzano. Without money, it's difficult to collect venom around the world, around all over Italy, and this is a problem. But in ideal project is this the one of the aim, to map the different venom composition in different region of Italy, because we know that French vipers or northeast or south viper difference in venom composition. And good point. And if you because one of the aim of EUVEN is the network. We need to work together, because we need, as clinicians, we need scientists and researchers need to clinical observation, because all these studies started from a clinical observation of the 1 patient, 2 patients that presented neurotoxicity. Yeah. And this is an example of translational research.
No, for sure. And it's good that this EUVEN network has been invented, set up to foster talking between these different parties. So no, thank you for making that point. A second question by Siddharth Bhatia. He says, you referred that the units, I guess, antivenom that were used are different from what is represented on the label of it. What unit of measurement do you use for reference? Is that correct?
No, probably is my mistake. In the unit I was referring me is the activity unit declared on the label by manufacturer, but different from one manufacturer to other manufacturer.
Yes.
And for example, there is a unit that is expressed by for example, in a Biomet antivenom is expressed by J.A, is a unit that is we can't understand. Is not comparable with other units.
Okay,
and
that's not very helpful.
one point. Yes.
All right. Another question by Marco Morano, and he says, "Thank you, David. I wanted to know if you have experience with French antivenom."
No, no, because in the past, Sanofi, that is the manufacturer of antivenom in France, use this antivenom only in France, and is on in theory is the best antivenom for us, because is manufactured against the Vipera aspis, but is expensive and is available for other countries outside France since 2 years. We have this antivenom, but it's not used at present.
Wasn't that also the company that stopped making antivenoms, or not? Am I mistaken?
Sorry?
Was that company, the French company you just mentioned,
Sanofi,
also the one that stopped making antivenoms for sub-Saharan snakes?
Yes, in past the Viper FAV was lacking during 2 years, and there is a paper that compare in this period the performance of Vipera aspis and Vipera tab, the use in Vipera tab in France.
Okay.
And the Vipera aspis seems to be better, but it's a French authors, and I don't know.
It's like me as a Dutch person saying something critical of a Belgium, which I would never do. IC wants to know, she says, "Thank you very much. It was a great talk, Davide. I wonder how effective anti-inflammatory drug administration might be compared to antivenom therapy. Could that help with symptoms?"
Yes, could help, but the problem is that when the patient envenomate jump the grade of severity, the local mechanism of inflammatory local mechanism started, and only antivenom may reduce the anti-inflammatory response locally if administered early. Then, when the inflammatory mechanism started, the anti-inflammatory treatment and corticosteroid, for example, may help, but don't stop the mechanism.
No, that makes sense. Even to a non-clinical biologist who studied physiology 30 years ago, like myself. It's a question that I see. It's a follow-up question. Can you distinguish which type of animal is involved in?
Okay.
Basically, yeah. Do you need is there a need for more guidance for clinicians to figure out what bit or stung patients?
Good. It should be good, but when the snake or spider is available now today with images is good, easy to send to experts and to recognize. The problem is when the spider of the snake is not available, and to correlate the clinical manifestation to animal bite, animal problems, or another reason.
Yeah, it becomes a mystery, a puzzle. I will do, if I'm not going to promise that we're going to get to all the questions, but I'm going to do a few more. We are still within the hour. Mark Zammit, who is a friend of yours, I know this.
Yes, Mark.
Yeah, Davide, thanks for a great presentation. As you know, many countries are reporting bites by exotic, foreign, non-native venomous snakes, which are increasingly popular as pets in Western countries. Do you see such cases in Pavia, and do you keep antivenoms for exotic snakes?
Yes. I have the slides about this because another occasion. No, we manage. There is also Catherine Faber that is connected, a friend from Zurich Poison Center, and that help, for example, in the past, because in Italy we haven't today stockpiling for exotic snake.
Mhm.
And in the experience of our poison center activity, we manage bite from Crotalus, from Atkistrodon bilineatus, Mexican moccasin, for example, from Vitis parviocula, that are very impressive because we are unfamiliar with this envenomation in Italy, but we have a network of clinicians. For example, our association, the European Association of Poison Center and Clinical Toxicology, EAPCCT, is a good point to start to have a network also between EAPCCT and EUVEN. And we ask for advice to other colleagues, but the problem is to have the antivenom. And the antivenom in these cases may be found in Switzerland, or in other countries. And is difficult because requires time, but through our network, we found the correct antivenom if available, and the correct amount of antivenom.
Yes. In the interest of time, I'm going to select a final question. I'm going to select a question that Leah Fitzpatrick is asking. And she asked specifically, have you had any cases of bites by the genus Steatoda, such as Steatoda nobilis? Because I think she's an island now, which those bites are becoming a problem there.
The Steatoda, yes, I have, we have had only one case of Steatoda, if I correctly remember, because he's a spider native of Spain. And we managed a case of bite by Steatoda that presented only local signs of envenomation. But the problem was to find a clinical information about this. Because sorry, just don't worry,
I'm not worried.
If I ought to, no, I have not this. Okay, no problem. Is an alien species in Italy, and there is some notes on the north of the Italy, and we have one case that presented only local signs that we, due to the lacking of clinical information, because there is only one case report, but written in Japanese, and this was difficult to understand, and the patient was observed for 24 hours to be sure that to avoid the clinical worsening of the patient, but only local signs were registered. But we have one case, one experience. Yes.
Okay.
Thank you for the question.
Let's hope it stays rare.
Right.
Davide, I want to thank you so much. It was an interesting talk, and thanks for being with us today. The audience, you've been great as well. I apologize for not having given everybody a platform for their questions. There's a few left, but that's always the case. So yeah, thanks again for being willing to give this interesting talk. I hope you will join future chats after the summer holidays as well, because we have a diverse program going. And all of you, I wish you all a good weekend. I mean, tomorrow's only Thursday, I know, but in the UK, tomorrow is a day off, and Friday as well. Today's my birthday, so within about 10 minutes, I will go to the refrigerator and get something to drink that is not Coca-Cola. So I wish you a premature weekend and a good summer holiday. All of you, be careful with these damned venomous creatures. They're really dangerous. Stay away from them. Thanks again, and I hope to see you in the future.
Ciao.